Hello, hello, hello. Hello, everyone, and welcome to another episode of Genetics Unsealed. I am your host, Fulabumi Adeleke, and today is a much promised episode coming today. It's a book review on Nessa Carey's Epigenetic Revolution. Now, the Epigenetics Revolution by Nessa Carey explains one of the most important developments in modern biology, epigenetics. Epigenetics is essentially the study of how genes can be switched on or off without changing the DNA sequence itself. At A-level biology, students learn that DNA contains the genetic instructions for building proteins. However, this raises an important question. If every cell contains the same DNA, why are cells so different? For example, nerve cells, skin cells, and muscle cells all contain the same genome, yet they perform very different functions. Carey explains that the answer lies in epigenetic regulation. Chemical markers attached to DNA and its associated proteins determine which genes are active and which are silent. In simple terms, epigenetics acts like a system of switches that control how genetic information is used. The book aims to explain these ideas in a clear way for non-specialists, whilst still describing the scientific discoveries behind them. And this book was actually was recommended to me by a former biology teacher of mine, as easy to understand. Around the turn of the millennium, the scientists completed the Human Genome Project, which determined the sequence of all human DNA. Humans possess around 3 billion pairs of base pairs of DNA inherited from each parent. Initially, scientists expected that understanding the genome would explain most biological traits. However, a major puzzle remained. Humans only have around 20,000 genes, which is far fewer than scientists predicted. This led researchers to realize that biological complexity does not depend only on the number of genes. Instead, it depends heavily on how genes are regulated. Carey explains this using a health analogy. Think of DNA as a library of instructions. Every cell contains the same library, where different cells read different chapters. A living cell reads instructions related to metabolism, while a neuron reads instructions related to electrical signaling. Epigenetic mechanisms control which chapters are open or closed. To understand epigenetics, it's important to understand how DNA is organized in the nucleus. DNA is extremely long. If stretched out, the DNA inside a single human cell will be around 2 meters long. To fit inside the nucleus, DNA must be tightly packed. This packaging involves proteins called histones. DNA wraps around histone proteins, forming structures called nucleosomes. Together, DNA and histones form a material known as chromatin. Chromatin structure plays a key role in gene regulation. Tightly packed chromatin makes genes harder for the cell to access. Loosely packed chromatin makes genes easier to activate. Epigenetic chemical markers influence this packaging. Carey explains two particularly important mechanisms: DNA methylation and histone modification. DNA methylation occurs when a small chemical group, called a methyl group, attaches to DNA. When methyl groups accumulate near a gene, the gene is often silenced, meaning it is not expressed. Histone proteins can also be chemically modified. These modifications change how tightly DNA wraps around histones. Some histone modifications activate genes, while others repress them. A useful GCSE level analogy is to imagine DNA as a set of instructions wrapped around spools. Epigenetic changes either loosen or tighten the wrapping, affecting whether the instructions can be read. One of the clearest examples of epigenetics is cell differentiation. During development, a fertilized egg cell divides many times to produce trillions of specialized cells. Despite having identical DNA, cells become specialized because different genes are activated in different cell types. For example, muscle cells activate genes for contraction proteins. Neurons activate genes involved in electrical signaling. Immune cells activate genes involved in pathogen defense. Yeah. Epigenetic markers ensure that once a cell becomes specialized, it maintains its identity. This process allows the body to produce complex tissues and organs. One of the most fascinating topics discussed in the book is how environmental factors can influence gene expression. Factors such as diet, stress, toxins, and early childhood experiences can affect epigenetic markers. Carrier describes experiments with rats where maternal behavior affected the stress response of offspring. Rat pups that receive more grooming from their mothers develop lower stress responses as adults. This occurred because maternal care influenced epigenetic changes in genes controlling stress hormones. This example shows that experiences early in life can influence biology through changes in gene regulation. Another controversial idea explored in the book is epigenetic inheritance. Traditional genetics states that inheritance occurs through DNA sequences, passed from parents to offspring. However, some studies suggest that epigenetic markers may sometimes be passed on to the next generation. For example, experiments in mice have suggested that certain learned responses can influence offspring behavior. This idea challenges. Hello? Hello, hello? Testing. This idea challenges traditional evolutionary theory because it resembles aspects of Lamarckian inheritance, where acquired characteristics are inherited. However, Curry emphasizes that scientists are still investigating this area, and that evidence remains limited. Epigenetic processes are also important in medicine. Many diseases involve abnormal patterns of gene expression caused by epigenetic changes. One of the most well-studied examples is cancer. Normally, certain genes called tumor suppressor genes prevent cells from dividing uncontrollably. In some cancers, these genes become epigenetically silenced. This allows cells to divide rapidly and form tumors. Scientists are developing epigenetic drugs designed to reverse these changes and restore normal gene expression. These treatments represent an important new direction in medicine. Now, moving on. Um, Within this book, Nessa Carey, she brought about some examples and historical discoveries to expand on the topic of epigenetics. One such was Waddington's trough. Now, although epigenetics is now a modern research field, the concept actually began in the 1940s with the work of Conrad Waddington. Waddington was interested in the major biological mystery, how a fertilized egg develops into a complex organism with many different cell types. He introduced the term epigenetics to describe the processes that link genes to development. To explain his idea, Waddington created a famous model called the epigenetic landscape. In this model, development is represented as a ball rolling down a landscape of hills and valleys. The ball represents a developing cell. The valleys represent a different possible. The valleys represent different possible cell fates. The path the ball follows determines what type of cell it becomes. For example, a cell could develop into a muscle cell, a neuron, a skin cell. The genes provide the basic instructions, but regulatory processes guide which developmental pathway is taken. The model was proposed long before scientists knew about DNA methylation or histomodifications, but it correctly predicted that gene activity must be controlled during development. Today, Waddington's ideas are considered one of the conceptual foundations of modern epigenetics. One of the most famous demonstrations of epigenetics involves the agouti mouse gene. In experiments with laboratory mice, Scientists discovered that mice with the same DNA could show very different appearances. Some mice were brown and slim, or healthy and normal weight. Others were yellow and normal, or obese are more likely to develop disease. The difference was caused by activity of the Guti gene. When the gene is highly active, mice develop yellow fur and increased fat storage. When the gene is less active, mice develop darker fur and healthier metabolism. Researchers discovered that DNA methylation near the Guti gene controlled how active it was. Even more surprisingly, the mother's diet during pregnancy could influence these epigenetic marks. When pregnant mice were given food rich in methyl-donating nutrients, their offspring were more likely to develop a healthy brown coat color. This experiment showed that environmental factors can influence gene activity without changing DNA. The Guti mouse therefore became one of the clearest examples of epigenetics in action. Another fascinating example of epigenetics can be seen in tortoiseshell cats. These cats have distinctive patches of orange and black fur. And interestingly, this pattern always occurs in the females. The explanation to this involves X chromosome inactivation. Female mammals have 2 X chromosomes, while males only have 1. If both X chromosomes were fully active, females would produce double the amount of certain proteins. To prevent this imbalance, 1 X chromosome in each cell is randomly switched off during early development. This process is controlled through epigenetic changes that silence 1 chromosome. Because the choice of which X chromosome is inactivated is random, different patches of skin activate different color genes. This produces the mosaic fur pattern seen in tortoiseshell cats. This phenomenon demonstrates that epigenetic processes can operate in an entire chromosome, the Dutch hunger winter. One of the most famous human examples of epigenetics comes from the Dutch hunger winter during the final months of World War II. During the famine, food supplies in parts of the Netherlands became extremely limited. Scientists started to study people who had been exposed to this famine while still in the womb. Decades later, researchers discovered that these individuals had higher risk of several health problems, including obesity, cardiovascular disease, and type 2 diabetes. Importantly, these health effects are linked to epigenetic changes affecting genes involved in metabolism and growth. The famine occurred during a critical stage of development, where epigenetic markers are being established in the embryo. This case study provides evidence that environmental conditions during pregnancy can have long-term biological effects. It also suggests that epigenetic changes may help explain how early life experiences influence health later in life. Some scientists have wondered whether epigenetic changes could influence evolution. Traditional evolution theory, developed by Charles Darwin, explains evolution through natural selection acting on genetic variation. However, epigenetics raises the possibility that environmental influences could also shape inheritance patterns. This idea resembles the earlier theories of Jean-Baptiste Lamarck, who proposed that organisms could pass on traits acquired during their lifetime. Although modern epigenetics does not fully support Lamarck's original ideas, some research suggests that certain epigenetic signals may persist for a few generations. Scientists are still investigating how common this type of inheritance might be. Another example of epigenetics comes from cloning. The famous cloned animal Dolly the sheep show that cloning is biologically possible. However, cloning often produces animals with health problems. One reason is that cloning requires resetting epigenetic markers in the nucleus of an adult cell, so it behaves like a fertilized embryo. This process is extremely complex, and errors in epigenetic reprogramming can cause developmental abnormalities. This demonstrates how important epigenetic regulation is during early development. The examples discussed in this episode show that epigenetics connects many different areas of biology. It helps explain how cells specialize during development, how environmental factors influence gene activity, and why identical DNA can produce different traits, and how some diseases develop. Epigenetics is also becoming increasingly important in medicine. Researchers are studying drugs that can modify epigenetic markers in order to treat diseases such as cancer and neurological disorders. As technology improves, scientists may eventually be able to precisely control gene expression using epigenetic tools. And together, the topics discussed today show why the epigenetic revolution by Nessa Carey is such an important work. She introduces readers to a field that is transforming modern biology. And by combining scientific research with real-world examples, she demonstrates that genes are not simply fixed instructions. Instead, gene activity is controlled by dynamic systems that respond to development and environment. This is a good book. I do believe that with some top-up, a GCSE student could read to understand the book, and for sure, an A-level student would be able to also. Although, I would like to add that you should most definitely read this book in one go. If you read it bit by bit, it is likely that you will forget some of the stuff that's mentioned, as a lot of the stuff, a lot of the things mentioned in this book are not in the A-level curriculum, or so the equivalent to that will be high school before college or university. And so it's best to read it through in one go, or at least in a short time frame, to avoid misunderstanding things and to better understand the book, if that makes sense. That's all for today's episode of Genetics Unsealed. This is your host, Fulwimi Aleshine Adeleke, signing out. I hope you enjoyed this episode. Bye bye.